Hello everyone, and welcome to Unit 4, our last unit for this semester. We're going to start off the unit talking a little bit about mechanisms of disease, which means spending some time discussing how exactly it is that these microbes cause harm to our body and cause disease, and also talk a little bit about how we diagnose disease, what the methods we use are to figure out what it is that's causing a problem in someone's body. Then we'll go into things like bacterial diseases, viral diseases, etc. But before I get into any of that, the first thing I want to do is just give you a little bit of information about preparing for exam 4, just give you some tips. Of course, you'll have a study guide as usual, but I always like to start the unit reminding you that this is a tough unit. There's a lot of information to learn, and to be able to be successful in this unit, you're going to want to start studying early, review often, and do a lot of comparing and contrasting. A lot of the exam questions are going to be ones where you need to be able to figure out of a list of organisms, which is the one that's causing the disease or which one is causing the problem in the patient's body. So to be able to do that, I really recommend you spend a lot of time comparing and contrasting these organisms with each other to make sure you are clear on what the characteristics of bacteria A are versus what the characteristics of bacteria B are. Of course, make sure you know the differences between the basic types of microbes. So unfortunately, sometimes at this point in the semester, people are still mixing up things like bacteria and viruses, or bacteria and protozoa, or protozoa and fungi, etc. If you need to, you may want to go back to Unit 1 and make sure you're very clear on the definition, the descriptions, the characteristics, etc. of our different types of microbes. You need to know the difference between a bacteria and a virus and a protozoa and a helminth and a fungus, and all of those other possible combinations. So you need to know the difference between a bacteria and a fungus as well, etc. Of course, make sure you know which microbe causes a certain disease. As you go through this unit, you'll be able to know that. It will say that on those slides, but just make sure you're clear that, Streptococcus pyogenes causes strep throat or streptococcal pharyngitis, and Bordetella pertussis, for example, causes whooping cough. So you'll want to know those types of things. And then again, you may need to review some main topics in previous units. You may want to go over the difference between gram-positive and gram-negative. You may want to go over the different structures of viruses, the different structures that we find in fungi, etc. So there's a lot to learn in this unit, and it may be time to review some of the things you learned in previous units, especially Unit 1. Okay, so now that we've gone over just a little bit about how to approach Unit 4, let's go ahead and get into this lecture. First, we'll talk about the mechanisms of disease, which really just covers the ways that microbes cause problems in our bodies, or the actual things that they do that causes damage. And then we'll talk a little bit about diagnosing disease, some of the procedures and techniques that we can use when a healthcare provider or professional needs to figure out which microbe or which specific microbe is causing problems in someone's body. On this slide, we are looking at the ways that bacteria and viruses cause damage to our bodies. I combined them on the same slide because even though bacteria and viruses are very different microbes, they're two totally different types of microbes, they actually do some pretty similar things. And when they get inside our body, there is at least a decent amount of overlap between the way that bacteria cause damage and the way viruses cause damage. So I have some different things listed here, and you'll see underneath each of the different bullet points, there's either one or two types of microbes under there. So basically, when we look at this one, when you say toxins are one of the ways that bacteria will cause damage in our body. Viruses don't really use toxins for the most part, but bacteria oftentimes produce toxins that are usually protein molecules, not always, but usually. And those toxins cause some kind of reaction in our body, whether that is cell death, or they cause us to produce more mucus, or they cause us to produce diarrhea. Whatever it is, this bacteria produces some kind of molecule, and that molecule causes our body to respond in some way or another, and in the process, our cells and tissues are damaged. For enzymes, both bacteria and viruses use various enzymes to degrade tissues. For both of these types of microbes, it is very common for them to produce or create some kind of enzyme that will interfere with our own metabolic reactions or start to break down our cells and tissues, and that allows the microbe to cause damage to the body and oftentimes further its own reproduction. Bacteria also produce a lot of different metabolic products or wastes as they go through metabolism. One of the things that we'll talk about in our bacterial diseases lecture is how sometimes during the process of something like cellular respiration or fermentation, some kind of acid or gas is produced by that bacterium, and that acid or gas ends up causing damage in the body. And then both bacteria and viruses are many times capable of causing what we call intracellular infection. So intracellular means inside of a cell. And although not all bacteria get inside individual cells and cause damage, many of them do. And then viruses, of course, as I hope you know by now, are obligate intracellular parasites, meaning they have to be inside some kind of living host cell to cause any kind of damage. And so both of these types of organisms will oftentimes get into individual cells, start to cause problems with metabolism, start to cause problems with things like cellular replication or DNA replication, and basically just end up killing the cell from the inside out. Another big way that bacteria and viruses cause damage to our body is actually not something that they necessarily do directly, but it is our response to them. Both bacteria and viruses tend to produce or result in a lot of B and T cell activity. So the thing about bacteria and viruses is that although, again, they are very different types of microorganisms, they are also both very different from the eukaryotic cells that make up our body. So our body is relatively good at recognizing these types of organisms as something foreign that needs to be attacked. And so when they invade, our body tends to produce a lot of B and T cells as a result. And that B and T cell activity can end up causing additional damage in our body. So this diagram here, don't worry at all about memorizing it or anything like that. It's just kind of here to illustrate some of the points that we're making here. And when we look at this infection, this is a bacterial infection, one that we'll actually talk about a little bit later. This bacteria comes in, releases some different enzymes, starts to degrade our tissues, and so that's one mechanism of action for causing disease. But in response, our body produces a bunch of T cells, a bunch of B cells, and all of their activities actually end up doing additional damage to our body. Sometimes, not always, but sometimes, our own body cells actually do more damage to our bodies when trying to fight off the infection than the microbe does itself. So oftentimes, disease process or the signs and symptoms that we experience are actually a result of our own body trying to fight the microbe as opposed to the direct damage the microbe is causing. Along that kind of same line of thinking, bacteria especially tend to activate the complement cascade quite a bit. And remember, the complement cascade is part of our innate defense. What happens there is a bunch of proteins are created, they join together, and they form a membrane attack complex that hopefully will destroy the bacteria by kind of poking a hole in it. But in this process, there is also damage that can be done to surrounding cells as well. Viruses tend to stimulate interferon production. Interferon production oftentimes will stop normal metabolic activities in cells. So again, just looking at this diagram here, this is just an example. Let's say we have a virus that has invaded this cell here. This cell recognizes that it has been invaded, and it starts producing interferon and releasing that interferon to other cells. And when that interferon gets into nearby cells, those cells tend to shut down their normal metabolic activities, and as a result, they stop functioning normally. And because of that, it starts to do damage to our bodies. And then finally, both bacteria and viruses tend to stimulate a lot of inflammation. So there's a lot of cells and a lot of fluids and a lot of proteins that will kind of rush to a particular area that has been invaded. And all of this activity, again, causes additional damage to our bodies. So not only is the microbe doing some damage, but our response to it causes additional damage to the area where the microbe is. Our next microbe is a prion. And prions, as you'll remember, at least hopefully from Unit 1, are infectious proteins. Basically what a prion is an alternate version of an already existing protein. So a prion is, if we say we have version A of a protein in our brain normally, a prion is like version B. So it's the same overall basic protein, just with some changes in the amino acids that are in there that change the overall structure of the protein. And basically what they do is our normal version of proteins help make sure our neurons function as they are supposed to and keep the same structure as they need to be able to do that. Prions, like version B of this protein, will come into our body. They will start to interfere with version A of that protein and kind of kick them out, cause all kinds of havoc there, where protein tangles start to form, the microtubules that will hold our cells together and make them structured and function the right way start to fall apart. And then overall, what that means is the membranes where these proteins were found, or the membranes of these cells, start to fall apart. What they end up doing is kind of folding in on themselves, and what that does is cause large holes to form in the brain. So if you look at the brain up here, that is a normal healthy brain, but when a prion gets inside our brain and starts interfering with the normal protein versions that we have, the cells and the cell membranes start to fall apart. They kind of fold in and like implode on themselves, and that leads to big holes being formed. The next type of microbe that we'll talk about is the fungus. Funguses cause damage to our bodies in kind of three main ways. The first one is toxin production. And the interesting thing about toxin production is that most of the time, the actual fungi that infect us, that cause direct infections to our body, don't produce toxins. However, there are plenty of fungi in the world that cannot directly infect us, but if we come in contact with their toxins or we ingest their toxins, it can be very problematic. So most of the time when we're talking about fungal toxins causing a problem, we're talking about a situation where some kind of fungus that generally doesn't directly infect the human body, but oftentimes will live environmentally, or maybe will infect a plant. You're looking at something called ergot of rye right now. So this happens on wheat and stuff too, but this one just happens to be rye. But anytime you see this kind of black looking kernel here, that is a kernel that's been infected by this fungus, and that fungus will produce a toxin. So in general, fungi that, again, bringing this back on track here, fungi that produce toxins are not ones that infect us directly, but they will do something like grow on the food that we're going to eat, or sometimes certain types of mushrooms will produce toxins and we consume them, or we consume some kind of grain that the fungus has grown on. We ingest the toxins in that way, and we start to experience a variety of symptoms, usually including something like nausea, vomiting, hallucinations, mood changes, potentially even up to things like going into a coma and dying. So fungal toxins can be rather mild, or they can be very, very serious and cause death. It really just depends on what specific fungus we're talking about, the amount ingested, and the type of toxin they have produced. In terms of the actual fungi that infect us directly, the ones that can grow on or inside our body, most of them cause damage in two main ways. The primary way really is through tissue destruction. Many fungi release a variety of different types of enzymes that will break down the proteins, break down the lipids, and break down other types of structures in our body that hold our cells together or that compose our cells. And so really what they kind of end up doing is getting on or inside our body and releasing these enzymes that kind of slowly dissolve us. And that's the primary thing that fungi do when they get inside our body. Of course, our body is also going to respond with immune function and things like B and T cells and potentially the complement cascade, etc. And so our body will do some additional damage to our cells and tissue as well. But really the main mechanism of pathogenicity for most fungi is they get on or inside our body and start releasing these enzymes that start to very slowly dissolve our skin and other types of tissues. Another big thing about fungi that a lot of people don't necessarily realize is that many people are allergic to fungi as well. So in addition to these skin and tissue degrading enzymes that they release, in addition to things like B and T cell activity that our body produces to try to get rid of the fungus, many people, not everyone, but many people are also allergic to these fungi. So there's an additional immune response there as well, with our body trying to respond to this invading pathogen and get rid of it. So that's another layer of damage that oftentimes happens when someone has a fungal infection. And then finally, we will talk about parasite pathogenesis. And really, I have grouped both protozoa and helminths into the same category here, because even though they are, again, different types of microbes, they oftentimes cause disease or cause problems in our bodies in very similar ways. So both protozoa and helminths oftentimes cause cell and tissue damage in two ways. One is physical damage with different structures they have, like hooks or barbs or suckers. You're looking at some great examples on this slide, and all of these happen to be helminths, but many protozoa have them as well. And basically what happens here is this microbe gets into our body and it uses some kind of structure that it has to just dig into our tissue, wedge itself in there, and start to cause destruction of our body in that way. Another big thing about these is they oftentimes use very similar nutrients to the ones that we use, and use them in very similar ways, because protozoa are not animals, but they're very similar in structure to animals, at least at a cellular level. And helminths are animals, which humans are too. So at a cellular level, there's not a lot of difference between a human and a helminth. And so both protozoa and helminths use nutrients in a very similar way that we do. They use the same ones. And so they oftentimes, once they infect the body, will just start to steal nutrients from the host. And most of the time in developed nations where people have access to food on a regular basis, this is not quite as significant. But for people who are food insecure, whether that is in a developed nation, because there is plenty of food insecurity in developed nations, or in a developing nation where there is oftentimes widespread food insecurity, what that means is the food that a child especially does get doesn't end up all going to them. At least some of it ends up going to this parasite that's living in them. And so when someone has very limited access to food, they have long-term infections, or they have other types of situations going on that make it just harder for them to live and to grow up and be healthy the way they need to. These types of organisms can make it even that much more difficult because they are stealing what little nutrients that person, especially a child, has available to them. Another big thing about the protozoa and the helminths, the parasites, as we like to oftentimes group them together and call them, is that they are very good at avoiding our defenses or altering our defenses. So they do things when they get in the body that make it so they can hide from our immune system, or they can evade it, or they can just kind of inactivate it. So there are different terms for this, and there are different techniques that they can use. They can use disruption, they can use evasion, they can use inactivation and suppression, and basically all of this kind of groups together in terms of saying that these organisms are very good at disguising themselves. They can kind of switch up the antigens on their surface so that even if we make antibodies to them, once they switch up their antigens, those antibodies don't work anymore, and our body has to make a whole new set of antibodies, and they just kind of keep going that way. So they're always basically one step ahead of our immune system. They can cover themselves in our own body cells or our tissues and make it so the immune system can't really find them. And then finally, a lot of times they will just produce different types of enzymes or other types of molecules that will inactivate our antibodies or just stop our ability, or at least slow our ability to make antibodies. So they're very good at avoiding our immune system. Okay, so now that we've talked a little bit about how these microbes actually do damage to our bodies, we're going to switch gears a little bit and talk about how it is that we can diagnose diseases, how it is that we can figure out what microbe specifically is causing a problem in someone's body. We can break up our methods or our techniques that we use to diagnose diseases into three main categories. So our first category is what we call the phenotypic method. And in the phenotypic method, what we're doing is we're using some kind of technique or some kind of method that focuses on the physical characteristics of the organism. We're looking at how much peptidoglycan it has, whether it has an outer membrane, does it have things like flagella, does it make endospores, does it make a capsule, does it have pili, etc. So we're looking at some kind of physical characteristic of the microbe. Our next method is the genotypic method. In the genotypic method, we are looking at what type of DNA or RNA, or potentially both, that an organism has. We're oftentimes looking to see what the specific sequence of the nucleotides is, what the specific sequence of the A's, the G's, the C's, and the T's are, because every type of organism on the planet has its own unique sequence of these types of nucleotides. And then finally, our third and last main category of diagnosing disease is the immunologic method. In this method, we are looking at the interaction between antigen and antibody, because antibodies are so specific in terms of which antigen they will bind to. If we get a match between an antigen and an antibody, and we know either what the antigen is or what the antibody is, then we know for sure that we're dealing with a certain pathogen. So we've talked about phenotypic methods a little bit on the previous slide, but just to go into some more details, we will cover them here. And phenotypic methods are actually ones that you guys had some experience using in the lab. Things like culturing, biochemical testing, staining, those are all things that you have done in the lab, And those are methods that are used every day on a regular basis to help identify an organism and figure out if it's the one causing disease in someone's body. So just some examples there that you guys have actually done using things like differential and enrichment media, things like mannitol salt agar, things like eosin methylene blue agar, things like McConkey agar, hectoin agar, all of those things, and plenty more of them. There are plenty more that you guys didn't get to use in lab. But all of those different types of media are ways for us to evaluate different types of microbes and figure out what someone's infected with. In terms of staining, we can do things like the gram stain, we can do an acid-fast stain, we can do a capsule stain, a flagella stain, etc. And all of that gives us a lot of information about the structure of the microbe, what its shape and arrangement is, what types of special structures it might have, like endospores or capsules or flagella, pili, etc. And so all of those staining methods that you guys did or that you guys at least looked at are great examples of methods that are used on a regular basis to identify microbes. And then finally, a last major method used in phenotypic analysis is what we call direct antigen testing. Now this one, I will admit, is a little bit of a gray area in terms of whether it belongs in phenotypic or immunologic, but most of the time it is grouped in phenotypic methods. So I kept it in that category because that's most of the time where everyone else categorizes it as well. But I do admit there's a little gray area here. Now, that all being said, with direct antigen testing, what we are doing is we are taking an antibody that has a fluorescent label on it. Basically, what we do is we create an antibody and we attach a little piece of fluorescent dye to it. It's fairly straightforward to do, nothing very special. Not a major procedure, pretty easy to do. And then what we do is we take that antibody and we add it to a slide that we have with our microorganism on it. So we'll just say that this is our smear. We have our bacteria in here, or other type of microbe. I'm just going to say bacteria for our example. So we add this antibody to our culture or our smear of bacteria on this slide. And then what we do is we take that slide, we put it under a microscope, and we check to see if it is glowing. If it is glowing, what that means is basically this fluorescently labeled antibody has found a match with an antigen on these microbes. And so since we know what antigen this antibody will react with, we know the identity of the microbe. So what we can do is we create this antibody, and let's say it's specific for Salmonella, or it's specific for Staph, or it's specific for E. coli. And if we think our patient has one of those infections, we take this antibody, we put it on our slide there, and if there's a match, then we have identified our organism. If there's not a match and we don't see any glowing, then we need to just use a different fluorescently labeled antibody, and see if there's a match for that one. In terms of genotypic methods, there are multiple ways to do this, but there are two very common methods. The first is using what we call polymerase chain reaction, or PCR. In this type of procedure, what we do is we take a piece of DNA that we want to make a bunch of copies of, and we put it into a special machine with special solutions that allow us to separate out the backbones of this strand of DNA we want and make a bunch of copies of those. So at the end of the process, this can take, it can vary in terms of time, but 4 to 6 hours, kind of at the minimum, is what you would need. It could go longer if you need to get a lot more copies or if you had very few pieces of DNA to begin with. But let's just say in about 4 to 6 hours, maybe 4 to 8 hours, you will have a bunch of copies of this initial strand of DNA. Then what you can do is start to analyze this DNA, figure out the order of the nucleotides, figure out the sequence of the genes in that piece of DNA or the A's, G's, C's, and T's, whatever it is you're looking for. And by doing that, you can start to identify the organism. You guys had a little bit of experience with this in lab this semester, most likely, when you did the viral identification experiment. When you loaded that gel and you waited a little bit of time and watched the DNA travel through that gel and were able to identify that organism based on whether it matched a certain known strain or not. So you didn't have a chance to do the PCR part. That part came to you already, but you did part of this overall genotypic method of analysis by analyzing that DNA and looking to see what the order of the sequence of nucleotides was and matching it up with a known sequence. Another very common method of genotypic analysis is what we call fluorescent in situ hybridization, or just most commonly abbreviated as FISH. FISH is another method where we use something fluorescently labeled. So what we do with this one is we take a piece of DNA that will match up with a target piece of DNA, kind of leave it a little bit open here, and we have our backbones, etc. And then these ones don't have a match. So we have part of the ladder has a kind of a full match and then part of it doesn't. What we do is we label this again with something fluorescent. Again, also not a very hard procedure, pretty straightforward and easy to do in a lab. And then we, again, will apply that to a slide where our organism is, or a sample where our organism is. And if the organisms on this slide or in this sample have a sequence of DNA that will bind to this part on what we call our probe. So this is called our probe. And if our organisms on this slide or in our sample have a sequence of DNA that will bind to this sequence right here, then we have what's called a match. It will start to glow under a microscope, and we will be able to tell what microbe was in this sample. Because again, what we can do is we can create a sequence of DNA that is specific for something like Salmonella or Staph or E. coli or some kind of fungal infection or a viral infection. Whatever it is, whatever we want to, we can create a sequence of DNA that will match the sequence of DNA in a specific organism that we want to identify. And then we can apply that to a sample. If we get a match and we see that glow, we see it fluorescing, then we know we have identified our organism. And finally, we will talk about the immunologic methods. You all probably did some of these in lab this semester again, or at least learned about them a little bit. Agglutination and precipitation are two very common methods. And with this one, with agglutination, what we're doing is we're taking a solution of antibodies that we know what they'll attach to, and we apply it to a sample of microbes that we don't know the identity of. And if we get them clumping together, then we know the antibody has attached to the antigen, and we know the identity of our microbe. This one is often done on some kind of plate or some kind of like paper-based slide. And what we do is we have our sample of microbe and we put it in there. And we take the antibodies that we know will match with a certain type of microbe. So let's say we are trying to determine whether our patient has Salmonella or E. coli. Those signs and symptoms that people develop when they get those infections can be very similar. So we want to know if our patient has Salmonella or E. coli. So let's say we're going to just start out assuming they have one of these two diseases. So we will create antibodies to Salmonella, and we'll just say those are the ones in blue here. We'll create antibodies to E. coli, and those are going to be the ones in green here. These are not very good antibodies right now. Sorry about that. They're not great, but they're there. So then what we'll do is we'll take the antibodies for salmonella and we'll put it on one sample from our patient. We will take the antibodies from E. coli and put it on another sample from our patient. Again, it's the same sample from the patient. We're just testing multiple possibilities at once here. And we will wait and see if we get some kind of reaction. If we do, what will happen is, let's say our patient's infected with E. coli. The sample of salmonella the sample that we were testing for salmonella will just look the same as it did before. Nothing will have changed. But in our one for E. coli, we will start to see, instead of just a normal kind of sample there, we'll start to see all this clumping form. And this clumping occurs because the antibodies that we placed onto this sample from our patient grab onto the bacteria, gather them together, and create what we call agglutination. Or they go through that process of agglutination. So they create these big clumps of bacteria, and we can start to see that. We can visually observe those clumps form. And when we have that visual observation of clumping, we can say that they were positive for this particular organism. Now our next one is precipitation. And with precipitation, what we do is use antigens to a certain microbe and look for antibodies that a patient might have. Most of the time, this is done in kind of a small agar plate. Usually it's round with 3 or 4 different holes poked in it, and then one in the center. In the center, we put a solution from a patient. So we take some of their serum and we put it in there because their serum will have antibodies to whatever disease they have been infected with. And in these outside wells, we put different solutions of antigens. My mistake. So we'll just say bacteria A, bacteria B, trying to get some different colors for you here, but it's not coming up. And we'll just say bacteria C right here. We'll ignore this whole for now, since I can't get the other colors to come. So what we do is let that sit there for a while. Usually we incubate it at something like 37 degrees, maybe a little warmer, maybe a little cooler, just depending on what we're talking about. And as we're waiting, the antibodies from the patient will start to diffuse out around where they've been injected. And the antigens that we've put into the little holes that we created will also diffuse through the agar. If there is a match between antigens and antibodies, we will see a line form between the patient sample and the antigens that we used. If there isn't a match, then everything just kind of stays clear in the area between the patient sample and the antibodies that we put. So in this case, our match occurred between bacteria A and our patient sample, so we can tell they were infected with bacteria A. And then our last common one that you probably didn't go over in lab too much, but is also still very, very common in laboratory situations, is what we call enzyme-linked immunoassay, or ELISA. With an ELISA test, what we do is we create a couple of different things. One, we're going to, again, create a fluorescently labeled antibody. Again, pretty easy to do, not a difficult technique. We're also going to take a plate, usually it's a plastic plate, and we are going to coat the bottom of that plate with a series of different types of proteins or antigens most of the time. What we're then going to do is we're going to take that fluorescently labeled antibody, we're going to put it into this solution that has those different antigens attached to the bottom. And we are going to look to see if we have found a match between the antibody and the antigen. If there's a match between the antibody and the antigen, the solution will turn color. So the basic principle, again, with ELISA is just we are taking a fluorescently labeled antibody and we're looking to see if the sample we're testing will have an antigen that that antibody can attach to. If the antibody can attach, then the thing will turn color. Now we can do this on a very large scale. Most of the time we use what's called a 96 well plate. It's just kind of this plastic plate that has 96 wells or 96 holes in it. And what we can do with that is we can take samples from 96 people and test them for the same thing at once. So let's say we want to run 96 different HIV tests or 96 different COVID tests or 96 different norovirus tests. Norovirus is a very common gastrointestinal virus that causes a lot of vomiting and diarrhea. It's the one you hear about on cruise ships a lot. So let's say we want to test a cruise ship and see if there's been an outbreak of those. What we do is we take a sample from 96 different people, 96 different people. We put each sample into its own plate, and then we can use this pipette that has multiple different tips on it, and we will expose the sample in each well to the fluorescently labeled antibodies, and we let it sit for a little bit. We check for color change. If in this case it stays pink, it was a negative. If it turns blue or purple, it was a positive. So we can just check and say, okay, patient number 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, they're all fine, but patient number 11, they were positive for norovirus. 12 was negative, 13, 14, they were positive for it, etc. And so we have a very efficient, relatively cheap, easy way to test a bunch of people at once to see if they were carrying whatever disease we were testing for.